you can see that, and I think Roger can see that at the same time. Okay, so this is chapter 8. And what I would say about chapter 8 is first of all, the title, of course. The title for chapter 8 is Chromosomal Mutations, Variation in Number and Arrangement. And mostly we're going to focus on the arrangement and when there's been, chromosomes have been broken and when they reassemble during meiosis and don't assemble correctly. Yeah, I don't know. There it is. Okay. So first up is some terminology that we need to know. And these, you'll see these terms in your book. They're in dark, like they're in bold print. And we've talked a little bit about aneuploidy before when we talked about Turner's and Klinefelter's, right, syndrome and Down syndrome, right? So aneuploidy is any time that you have the normal number of chromosomes plus or minus 1, usually, okay? So monosomy would be like a case with Turner's when you're minus 1, you're 45 chromosomes instead of 46, you only have 1 X. This is normal, dysomy, I don't know why they put that on there. And then trisomy, like Down syndrome, is trisomy 21, that's 1 extra, right? You can have just individual extras, but that's super, super rare. Those fetuses usually are not carried to term. So that would be something that would be found in like a miscarriage in tissue, right? Not so much in a live person. Now, diploid we're familiar with, of course, because that means 2N, right? The one that I want you to focus on is polyploidy. This one. Polyploidy technically means an extra set of chromosomes. So we say aneuploidy when it's an individual chromosome or maybe 2 chromosomes or 3 chromosomes. But if you have a whole extra set, we call you polyploid. Where the set originates from is where these prefixes come from, autopolyploidy and allopolyploidy. Those are the 2 that I want you to take a look at. Autopolyploidy, multiples of the same genome. So obviously this isn't going to happen in humans, but it will happen a lot in plants where they'll have a whole extra set of chromosomes and it will be from the same species, meaning for whatever reason, the chromosomes didn't separate during meiosis, right? So their sex cells ended up like a pollen grain, ended up with a whole extra, ended up 2N, and then it fused with an ovule, and then it was 3N, right? Allopolyploidy is when you have multiple sets of chromosomes from a closely related species. So this would be like, you know, like red wheat that we eat, we grind up for flour. That is a polyploid. It's actually, I think, a tetraploid. So I think it's 4 different closely related species of wheat that have been cross self-fertilized multiple times to give us the wheat that we have today. All right, so the next slide is some examples of how that might happen. In general, we refer to it as non-disjunction, meaning the chromosomes, the sister chromatids did not separate properly, right? So you can have a first division non-disjunction. So in meiosis 1, and you would have a cell that still had all the tetrads and another cell that had nothing, right? That's going to be a problem. That's going to be a monosomic individual gamete, right? Or that's going to come from the other part, the other sex, right? Or, and then these gametes, when they're fused with a haploid of their same species, are going to be trisomy. They're going to have 3 of the same chromosome. You can also have disjunction at the second, like I would say, prophase 2 of meiosis, well, anaphase 2 of meiosis. And we would end up, this part would be normal, so we would still have a chance of getting a normal offspring, but then we have one cell that has twice as many chromosomes, and they're going to get a third from the opposite sex. And you can also end up with one that only has one from the opposite sex. So these are just to show you some of the different ways that you can end up with aneuploidy. Now, monosomy is usually not tolerated in humans or other animals, although I would say fruit flies, some other insects, and maybe some reptiles get away with it. A lot of times, even if the organism survives, having only one copy of the gene to make into protein is insufficient. And there's a word for that. It's called haploinsufficiency. Because it's haploid, right? I don't know how to spell that. So and haploinsufficient. All right. Click. I don't know what this is supposed to be a picture of. This is a picture of Jimson weed, and this is 12 possible trisomic alteration or conditions. So this trisomy in this one happens a lot. Here's Down syndrome. These girls are siblings. And this one she has an extra copy of 21. And some of you had that for your karyotype exercise that we did, right? This is showing the increase in Down syndrome with time that the eggs are waiting before they get fertilized, right? 20, 25, 30, between 30 and 35, it jumps significantly. I think this is 1 in 800 or 3 in 1,000, and it jumps up significantly as the older the mother is to 1 in 30, and as common as 1 in 15. I always tell people that's why my parents were so excited when I was born, because they were 46 and 47. My mother used to always say, like when I would whine, like when I was in high school, and I'd be like, oh, I don't like my nose and my hair is so ugly. My mother would just be like, you're normal though. You're just normal. And when I was in high school, I had no idea why that was, a thing. But later I was like, oh, that's why she used to say that. Okay, I don't know. This is a chromosome 13. So this is a trisomy 13. And these are all of the conditions associated with that, right? So 47 chromosomes, 3 copies, blah, blah, blah. Okay. Cotton. Okay, let's start talking about some of the chromosomal rearrangements. I'm going to try to keep it as simple as possible. I think I will mute that for a minute and stop sharing for a second. Why can't I do that? Let me make this bigger. There we go. Okay, so we're going to talk about 3 types of chromosomal mutations where chromosomes, like I said earlier, where chromosomes are broken, and when they reassemble, they don't assemble correctly. Okay, so almost always we refer to these as rearrangements. And I would remind you that they're going to be happening as a result of crossing over in prophase 1 of meiosis. And the main causes of this is when both strands of sister chromatids break and the wrong ends reconnect, so you get a whole shuffle. Sometimes there's errors in crossing over, and the chromosome actually continues to make a copy of a piece that has gone missing. And crossing over increases a lot when DNA sequences repeat. So if a person has a lot of DNA repeats, they're going to have more likely crossovers. And then when they cross over, chromatids cross over, it's going to be less likely that they're going to go back to the correct sister chromatid. Okay, so first type is what we call a duplication. An easy one. So a part of a chromosome, a segment, not the whole thing, but a part of the chromosome is copied twice, ends up on one of the sister chromatids twice. Picture of that. So part of the chromosome, part of the chromosome. Okay, so I have a picture here from our book about a duplication. So let's go back to this. I think this is a, I said, yes, please share that. There we go. Go away. Okay, if we take a look at this one in the center. Oh, sorry, you can't see that. I'll remember. So here's one sister chromatid, right? And they just labeled the segments A through G. When there's been a duplication, in this particular case, B, C, and D are now twice, right? We've got BC and we've got BC. Oh, I guess it's just BC, no D, right? Can you see how this sister chromatid is now significantly longer, right? And we have those extra pieces, right? Those extra genes. Now, what happens? Sometimes this is a problem because of the increase in length, the tetrad won't be able to form properly. But also, usually it leads to, in the phenotype, there's either too much or too little of a particular gene product. This is the picture I want. I can just, I think, we mutate that. I have to stop share so he can see. Two effects that we have to pay attention to. The first one is that you have, so they say like gene dosage imbalance. So meaning too much of one protein, maybe not enough of another protein, depending on which one of the chromosomes, one of those sister chromatids get into the organism. This can, if it affects a particular developmental pathway, obviously it's going to have more of an impact. But a duplication is really all-star good for evolution. We know there have been hundreds and thousands of gene duplications, and why is this good? Because what happens is the original gene can remain functional and the duplication can mutate. And mutation, right? That means that's the source of variation, and variation is the basis of evolution by natural selection, right? So if you don't have any variations or any new variations, you're not going to get any change over time. And the example that I have is the globin genes meaning for fetal hemoglobin versus adult hemoglobin. Okay. So those have a lot of genetic similarity, and it's pretty much established that adult hemoglobin is a duplication of fetal hemoglobin, and it is mutated, and so you can breathe air instead of breathing air out of water or having to be in amniotic fluid your whole life. So that's an example of how that worked. Okay, now a not so happy example is a deletion, another type of result of chromosomal rearrangement gone awry. And if you think about our chromatid that got the extra BC, that means that its sister chromatid now doesn't have the genes B and C, right? Whoops. And what are those really important? Then that fetus may not develop, or there may be missing things, right? Just like it sounds. You've got missing pieces. In order to be able to see that missing pieces in a karyotype, it has to be about 5 million base pairs. It's kind of a lot, right? To be able to see it with a microscope. If both of the chromosomes if both of the chromosome copies in each of the parents both have deletions, it'll be lethal. Okay. So if both chromosomes have the deletion, and then I also wrote, you can get a lot of multiple defects in the organism, even if you have one normal chromosome and one that's missing the pieces, because as we talked about up here, the gene dosage imbalance, too much of a protein or not enough of a protein, right? If the deletion is in a gene that is recessive, and you got a good copy of the gene from your other parent, you might be okay, right? You might have, and your other copy from your other parent is normal, or wild type, right? That might be enough, even though it's not as much as you would like to be able to make, even though you don't have like a backup copy of that gene. And again, like we talked about, if sometimes this is not going to produce enough protein. I'll just put haplo insufficient. Now that I know how to spell that word, we're just going to use it for all of these examples. No, just kidding. Okay. There are some examples in humans. Deletion of part of chromosome 5 causes, I'm not going to say it the French way because I don't know how to pronounce it in French, but we say cry of the cat because the cry of the baby sounds like a kitten crying. There's examples of a deletion in chromosome 4 that gives a different syndrome, and chromosome 7. All of these usually result in the death of the child from between weeks of age to 3 years in age. So even if they survive, it's only for a short time. So let's take another look at that. Okay, just share. Stop doing that. Sorry. I know. If I talk to the computer, it's not going to make it do what I want. I know. But let's look at, and then you guys can't see. Let's look at the deletion. So in this case, looks like we're missing, so A, B, C, we're missing D. So it's a short deletion. Okay. The last thing that we're going to talk about, we're not going to talk about reciprocal and non-reciprocal translocations. I'll give you some definitions, but that's it. In an inversion, which is the last example up here, I'm going to title it number 3. This again is just like what you expect. It's when a piece of the chromosome has flipped and inverted 180 degrees. So this requires 2 breaks in the chromatid, right? So you don't lose any DNA, right? They're still the same size, which is great. But they're not, the genes aren't in the right order. And a lot of times the order of the genes is important for gene expression. This is also going to impact, I don't know if I have a picture of that, but it will also impact when this chromosome is copied again. It will have a hard time forming, it'll make a loop, and it will have a hard time forming a complete chromosome, like for the next rounds of mitosis, right? Because the parts aren't going to match up. Let's see the inversion, I think. Let's see if I can find deletion loop, those guys. So here's an, I think this is the inversion picture. That's actually unequal crossing over, sorry. I guess I don't have that picture. All right, anyway, I'm just gonna let you look at this picture for a minute. This one. This one. So this looks like an original that where we actually have a deletion. In this instance at the top, it's a little piece of A that gets lost, right? So one of the sister chromatids is intact and the other one is short and A. And so when that is replicated, sometimes because the the centromeres can't align up correctly, you end up losing even more of the the chromosome, right? So you can see that a little bit down here in this one. Right, so now we've lost both C and D, and now this chromosome only has A, B, and E and F. Right. Okay. Let's mute that and stop sharing. Some more words for the inversion. Okay, so 2 breaks in order to flip 180 degrees. And if you only have, if both copies of the chromosome invert, usually that turns out to be okay. The person turns out to be normal because they can still form a meiosis tetrad, and you don't get those loops where you get pieces broken out. If you are heterozygous, though, if you only have 1 copy of your chromosome that has inverted, then you're going to have those inversion loops that are going to form because the centromeres have to line up, and the only way they can line up is if some of the chromosome is like extruded or put into those loops. And then there are versions of this where the centromere gets stuck in the loop, and when that gets cut out, now that chromosome is lost entirely because it doesn't have a place to connect the kinetochore to the spindle fibers, And so in the next round of mitosis, it just gets lost and you're missing that chromosome. Okay, importance in evolution. Inversions often suppress more recombination. And the other thing that can happen is, on the downside, is that many genes only function positionally on a chromosome. And so if you change where they are on a chromosome, that's going to cause a problem for the gene to actually be expressed. All right, there's one more. There's 4 types of rearrangements. The fourth is called a translocation. And that is when pieces of either homologous or non-homologous chromosomes trade parts. So don't, it says translocation is between non-homologous pieces, sorry. Pieces of non-homologous chromatids cross over and exchange pieces. When we drew pictures of meiosis on the board and I talked about crossing over, I drew pictures like this, where we had 4 copies of chromosome 1. And I said, okay, these are plain chromosome 1, right? And these are filled in chromosome 1. And this would be a homologous pair crossing over if we had like a crossover here, right? So what we're talking about in this case is if a piece of chromosome 1 gets traded with chromosome 12, and a piece of chromosome 12 comes to chromosome 1. That's usually not good. Oftentimes the centromere is lost. We talked about that. If the break is in the middle of a gene, obviously, then that gene function can be lost. And I put something else too. That's all I got. Sorry. All right, so almost there. Just 2 more little things, I think. I'd like to show you guys. So let me do share screen again so that Roger can see better. So this is a person with, I think they've marked the little missing piece of chromosome 5. It's a cry of the cat. And then I wanted to show you bar eyes is a deletion mutation, I think, in fruit flies. Yeah. And then this is how complicated it can get, right? So the 2 blue chromatids and the 2 orange ones. So this would be in the offspring when they're trying to go through meiosis, and the centromeres can't align, and the genes, the only way the genes can line up with each other is if these loops are formed. And then frequently what happens is when those loops are formed, that gives a lot of extra stress to the chromosome and it breaks, right? And that's when things happen, like this one's still normal across the top, but this one gets a break here. And we get this crossover missing a piece, this one missing a piece, and this one a combo of the two of them. And so that's when you get a lot of non-functional proteins made, right? That's a Robertsonian thing. That's not important for us. Okay, so I want to show you this picture. This is a particular different chromosomal mutation that's called Fragile X syndrome. And these are areas of a chromosome that are more likely to break. There are many common fragile sites. There's about 100 in every human. But there are some rare fragile sites, and Fragile X is a rare one. Okay. It's present about 1 in 5,000 live male births, and it's caused by multiple copies of the repeat CGG. And remember I said when there's a lot of repeats, that's a lot of times where chromosomes break or try to cross over. So that's Fragile X. I don't know what all that nonsense is. Okay, I think that's the end of sharing his stuff. I think that's the end of what we need to talk about for this chapter. See, that wasn't so bad, was it? No. All right, so I'm trying to look through my notes real fast and see if there's anything else that is important. I think we're good. Questions, comments, worries, concerns about the pending doom?
Did you say look at the handout? What do you mean by that?
So there's
Sorry.
In Canvas, I put this here for you. I don't know, maybe I can't open Canvas at the same time.
Yeah.
Probably not. When you look in Canvas, in the module, I put a new module up today, module 4. There is a lecture video for chapter 7, Roger, for you, because I didn't record it, but it's recorded from before, and it's the chapter 7 from this book. So it's current. And those of you that have already forgotten what we talked about in chapter 7, that's available for you as well. And then there'll be this video for chapter 8. And there is a handout for chapter 8, and it's labeled chapter 8, chromosome variation, spring 2020. But minus the first part of the handout, it's all pretty much stuff that we've just gone over or we've gone over before. Okay, so like I think in the handout, it talks about amniocentesis, and we talked about that before. And it talks about Turners and Klinefelters as aneuploidy, and we've talked about that before. We talked about that in chapter 7. So it, with minus that beginning section of the handout, it's pretty good to study from. That's what I'm trying to say. Right. Okay, I'm going to stop the recording.